Ever wonder how we figure out if something can actually, like, fight off those bad bugs? You know, the ones that can make us sick.
Yeah, fascinating stuff.
Well, today we're doing a deep dive straight into some lab techniques that are used for just that.
Okay, sounds good.
And the best part is we're breaking it down without getting too bogged down in all the crazy technical details.
Right.
So we're talking about Agar diffusion assays.
Uh-huh.
These are basically clever ways to see if a substance can stop microbes from growing.
Yeah.
Our deep dive is based on a whole bunch of information about different types of these assays.
Okay.
And they're super common when scientists are trying to suss out, how well something works against those microbes.
Right.
The basic idea here is that we're looking at how a substance spreads through this gel-like material called agar, which has microorganisms on it.
Yeah.
And get this, if the substance is strong enough at a certain point, it'll actually stop those microbes dead in their tracks,
Yeah.
creating what's called a zone of inhibition.
Zone of inhibition, yeah.
And the size of that zone can give us a clue about how effective that substance is.
Right.
But it's not just about the size. It's also about how well the substance can spread out.
Exactly.
It's a bit of a balancing act.
Uh-huh.
Basically, we're talking about the techniques behind how we discover and test new medicines to fight infections.
Yeah.
So let's jump right in.
Okay.
We've got a few different methods to unpack here.
Okay.
First up, we've got the well diffusion method.
Right.
So what's the deal with that one?
Well, with well diffusion, you're basically cutting little holes or wells into the agar.
Oh, interesting.
And these wells are filled with liquid samples of the antimicrobial agent we're testing.
Okay.
And as the agent spreads out from the well, if it's doing its job, you'll start to see that clear zone forming.
The zone of inhibition, right?
Exactly. It's a great way to test liquids directly, and you can even compare different samples or different strengths of the same substance all on one plate.
That's pretty handy.
Yeah, it is.
But I imagine there are some limitations too, right?
Well, it can be a bit of a manual process,
Uh-huh.
and the results are mostly about comparing those zone sizes rather than getting super precise measurements.
I see.
So it gives you a good idea of relative effectiveness, but it's not always about pinpointing exact numbers.
Got it.
Yeah.
Now let's move on to the agar plug diffusion method.
Okay.
This one sounds kind of cool.
Yeah, it's a good one.
It's all about seeing if one microbe can produce something that can actually harm another microbe.
Right.
So how do they set that up?
Well, they take a little piece of an agar with a producer microbe growing on it.
Okay.
This little piece is called the plug,
Uh-huh.
and they plop this plug onto a plate with a test microbe.
So it's like a microbe showdown.
Yeah, kind of.
So what happens next?
Well, if the producer microbe is making something that's antimicrobial, you'll see a clear zone popping up around the plug.
Oh, because it's stopping the test microbe from growing.
Exactly. It's a pretty neat way to spot those naturally produced antimicrobials.
That's fascinating.
It is, isn't it?
Like nature's own little weapons.
Yeah.
But I'm guessing there are some limitations here as well, right?
Yeah, for one, you have to grow 2 sets of microbes, which can be a bit more work,
Uh-huh.
and it's more of a qualitative assessment.
Meaning?
You can see the inhibition happening, but measuring exactly how strong it is can be a bit tricky.
Right.
Yeah.
Okay, on to the next one, the spot assay.
All right.
This one sounds like it's all about speed.
Yeah, it's a really quick way to check a bunch of samples at once.
So how does that work?
Well, you basically put tiny drops of the test solutions directly onto the agar surface.
Okay,
And if there's inhibition happening, you'll see clear zones under or around those spots.
makes sense.
It's super simple, uses very little sample, and is perfect for a quick first screening.
So it's like a quick and dirty test
Kinda, yeah.
to see if something's worth investigating further.
Exactly.
But I bet there's a trade-off.
Well, the main thing is that it's mostly a yes or no kind of result.
Uh-huh.
It doesn't give you a ton of detail about how much inhibition is happening.
And it might miss some subtle effects.
Yeah, it's not as sensitive as some of the other methods.
Okay, last but not least, we've got the cross-streaking method.
Right?
This one seems to be all about how different microbes interact with each other.
Yeah, it's a visual way to see if 2 types of microbes are basically getting in each other's way.
So how do they do that?
They streak one microbe on the agar and let it grow a bit.
Huh?
Then they streak the second microbe across that first streak.
Like they're crossing paths.
Exactly.
And what are we looking for here?
Well, if the second microbe's growth is stopped where it meets that first streak, it means they're not getting along.
Oh, so one microbe is inhibiting the growth of the other.
Exactly.
Interesting.
It's a simple way to see those microbial interactions playing out.
But it's probably not perfect, right?
Well, it's really just a visual check. Hmm.
It doesn't tell you how strong the effect is, just that it's there.
I see.
And it only works for those specific microbes that you're testing.
Right, so it's limited in scope.
Yeah.
So let's recap what we've learned.
Okay.
We've got, well, diffusion for testing out liquids,
Uh,
agar plug diffusion for seeing those secreted antimicrobials,
right.
the spot assay for a quick screening of a bunch of samples,
Yeah.
and the cross-streaking method for observing those microbial interactions.
Exactly.
It's clear that these agar diffusion assays give scientists a whole toolbox for tackling those questions about antimicrobial activity.
They do, yeah.
And each method has its own pluses and minuses, making some better suited for different research situations. But one thing that ties them all together is that clear zone of inhibition.
Right.
The presence and size of that zone are key indicators of antimicrobial potential.
Exactly.
It's pretty amazing to think that these seemingly simple methods are actually at the forefront of the fight against microbial threats.
Yeah. It really makes you appreciate the power of basic scientific techniques, doesn't it?
It does.
So until our next deep dive, keep thinking about that invisible world all around us and the amazing ways we're figuring it out.
Great stuff.